From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 1st, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 52nd episode of Matters Microbial. I am so very grateful to each and every guest on this podcast, and especially to each of you who listen and or watch this podcast. It means a lot to me. First, a bit of humor. I have never visited Yellowstone National Park, although that is a blemish on my microbial soul not to have done so. A friend of mine recently went, saw this sign near a thermal spring that says not to mark a microbial mat, and thought of me. Punctuation can matter. Next, a couple of viewers of this podcast asked about the odd things around me in my office. Here is a handmade computer buddy, complete with hat, that a former student and researcher, Kat Schmidt, made for me. It's here for every episode. My student research lab is getting ready to transition to the fall semester. First, Doc Marcia Hannah gave a great presentation of her work with thiotropic bacteria yesterday. Doc Marcia Amanda continues her work isolating and characterizing marine bacteria that degrade agar on Petri plates. Most of the time, we see nice pitting from such colonies, but Amanda brought this oddly variegated colony to my attention. Is the colony phenotype unstable? Are there hitchhiking microbes? Time will tell. We've explored several aspects of the human microbiome in past episodes of this podcast. It's a fast-moving and complex field, which is no surprise, as the human microbiome is as vast and various as the Amazon rainforest itself. Researchers have begun to uncover relationships between changes in the human microbiome and certain diseases, as this review paper illustrates. And in some cases, as seen here, even issues revolving around that terrible suite of diseases that we call cancer. But it is certainly a very complex topic and difficult to distinguish between correlation and causation. So I am beyond delighted to welcome Dr. Justine Debelius of the Department of Epidemiology at Johns Hopkins Bloomberg School of Public Health to discuss one such example relating a part of the human microbiome to nasopharyngeal cancer. Justine will also tell us about her journey into the microbial sciences. Justine, welcome to the Quality Quorum for today.
Thank you so much for having me. I am so excited to be joining you.
On episode 52. So here we go.
Fantastic.
Would you like to take a moment to talk a little bit, just briefly, about how you got into the microbial sciences?
Sure. So there are two stories. So one is that as an undergraduate, I got work study, and my work study opportunity ended up being in the medical microbiology lab at my liberal arts college. And so I spent 4 years making agar and autoclaving tubes that had been used. And that was my, and I think I inventoried her freezer. My microbiology teacher had an amazing collection of staph isolates from the community. And after I left undergraduate, I went to grad school. And my first year of my PhD, I was doing rotations, and I thought I was going to work on cell signaling in humans using fluorescence microscopy. And it turns out that I don't have great lab hands. And I, in fact, was trying to make an SDS page gel. And SDS, sodium dodecyl sulfate, is like a detergent. And I was walking either to my bench or back to the lab cabinet with this big jar that was 3/4 the way full of SDS. and I stumbled, and the SDS went everywhere. And it was like Tinkerbell had sprinkled SDS all over the lab. And so after that, and several failed PCR attempts, some of the older students in the program came to me and said, Justine, there's a computational lab upstairs. You should go talk to Rob Knight. So I went and I rotated with Rob, and Rob very graciously accepted me into his lab, and I've been doing microbiome since then.
Well, it's interesting to tell everyone that there are, the term in vivo, where we study living things, in vitro, where we study the enzymes. And even though it just hurts me to say it, there's this term in silico. In other words, studying things simply on the computer. And there's a whole world about this. And it's easy for experimentalists of the old school to not appreciate what's possible with it. But boy, has it changed the field.
I would like to claim that I still have some experimentalist cred. I do my experiments with data, but I was trained as an experimentalist, and I think I still, sometimes to the frustration of some of my colleagues, work through problems like an experimentalist.
Yeah. I mean, it's really interesting in my own lab here when we're looking for a homologue of a particular gene. And we'll design primers around it, and we'll PCR that up and clone it and then put it into a cell to see if it's expressed. And that's a nice example of how you can make a hybrid of that approach. But I think the point that I want the listeners and viewers to understand is there are so many paths in the microbial sciences today. I actually have started to say, instead of microbiology, the microbial sciences for that reason. So I think a good thing to start off with, because I know you have some data to talk about, and I know you have some terminology to talk about. And from time to time, I'll ask you to kind of clarify that for folks who are, and as you may or may not know, my wife's a mathematician, so I often describe myself as being innumerate, but I can do arithmetic. So there we go. So I think a place to start is maybe a lot of people have talked about the relationship between the microbiome and any number of phenotypes. And there's an awful lot of, I think, inaccurate or over or underselling that we see from the media itself. And that's one of the reasons I'm excited to have you here today, because everything of yours that I read suggested to me you were extremely cautious and careful about the analysis that you're doing and what it might mean.
I try to be. Most of the work I have done has been association, and I want to be clear about the fact that the work we're doing is often association. There are studies you can design that would be prospective studies, so looking forward. And in fact, one of the reasons I did a postdoc in Sweden at the Karolinska, and one of the reasons that I wanted to go work at the Karolinska, is that they had a collection that they haven't quite finished sequencing of fecal samples that are linked to population records. And so in a decade, they'll be able to go in and say, if I took a fecal sample from Mark when he was 60, does he have Parkinson's disease when he's 70? If I look at Justine's fecal sample from when, these are all a cohort of 60-year-olds, but if I was in that cohort and I look at Justine's fecal sample, does she have breast cancer in 5 years? And so I think that there's a lot of power there, but I think that there are also a lot of challenges with those epidemiological studies. Emily Voigtman at the NIH does some absolutely amazing work on this topic. And she's kind of who I look up to in the cancer microbiome field in how to do this well as both an epidemiologist and a microbial scientist.
I think I'm really glad that you put this in here as a proviso for people listening and watching, because it's easy for things to become, and you know, I don't want to say exaggerated. We've all had the experience as scientists of really wanting something to be true, and you have to really restrain yourself. You know, like if I do it one more time, maybe the experiment will pay off. No, if it's not replicable, it's not real, you know, that kind of thing. And when you talk about something as complex and various as the human microbiome, it's amazing. So
Yes.
perhaps a good place to start is the model system that you're going to be talking about today, and it's it's something that I didn't know very much about. So could you please tell the listeners and viewers about nasopharyngeal cancer?
Sure. So I worked on a study of nasopharyngeal carcinoma. So the nasal pharynx is up inside the top of the nose, back behind where you're COVID swabbing when you think you're going to reach your eyeballs. That's approaching the nasopharynx. And there are sort of two types of NPC. So there's a type of NPC that you see all over the world. And if you look at this map of the distribution of nasopharyngeal carcinoma around the world, you'll see that there are a lot of blue areas. And then there are a handful of red areas, and those have endemic NPC, and that's what I'm studying. And endemic NPC is actually a virally associated cancer. And so in those areas, Epstein-Barr virus is associated with nasopharyngeal carcinoma. And the interesting thing about Epstein-Barr as a virus is that 90% of the adult human population carry EBV. So typically we get it from our parents as young children. If you get it as, you know, a teenager in the West, you might get mononucleosis. But if you live in this endemic area of China and you get it from your parents, it might put you at risk for nasopharyngeal carcinoma down the line. And so it's this, you know, it's common in southern China where we looked. It's less common other places.
So when you hear about something like this, and again, it's something where when this begins to be talked about, folks will become nervous and upset. But there are many risk factors for all kinds of things that we talk about. And at the same time, there are other risks that are far more frightening that we take for granted every day, like driving.
Absolutely.
Mm-hmm. So it's interesting why there's this elevated, and I hesitate to assign a number for the elevated risk factor in this area of southern China, but it's certainly something that shows up statistically and epidemiologically. So there must be some other factor taking place there.
Absolutely. And I did my work as part of a larger case control study, the NPC-GEE study. And so they collected from, I believe, 9 sites. And when people came into the hospital as a new NPC case, their doctor would refer them to the study. And they would be brought in for an interview with a trained field worker who would ask them all sorts of questions. They would ask them what they did for a living, what kind of food they ate, did they use tobacco products? And if they used tobacco products, how much, how long? I think they went through a dental exam with a dentist, although they may have just asked about dental health. And then they collected a sample of an oral sample from all the participants. They then went to the population register in a matched area of China, and they found age and sex matched adults. And they invited them to participate in the study too. And they asked those adults the same set of questions. Mm-hmm. And so if you didn't have cancer, they asked you about your oral health and about your profession and about your diet. And based on that design, we're able to say, if you have cancer, you know, did you smoke 5 years ago? If you have cancer, do you have more dental disease? If you have cancer, are you eating certain types of food now, or did you eat certain types of food as a child that are maybe associated with the cancer risk?
Now, this is very important. First off, you don't need my approval. It's very well explained there. But I wanted to say that it's very typical in kind of, there's a saying about the press: if it bleeds, it leads, and that's been around forever. But I would say anything that's kind of outrageous is what we seem to go with in the media. So when you hear about a horrific disease or someone who's had something terrible happen to them, and there's one other aspect of their life, doesn't matter what it is, people try to connect those two things. So what's really important for the listeners and viewers to understand here is that what you just described is a way of trying to suss out whether there's something specific leading to this particular disease.
Absolutely.
Right. And that led to perhaps some ideas of what was living in this particular area so were you, I hate to use the word swabbing because that comes to swab story, which always makes me laugh, but instead of sob story, how many times have you seen an article that says bacteria were found in, and like your shoes or the bathroom or whatever? So I always call those swab stories, and I do my share of swabbing too. But the point is, so you were looking in the nasopharyngeal area to see what kinds of microbes were there in both of those groups then?
Yes. So the saliva samples were collected because several people wanted to look at the Epstein-Barr virus that was there. And I consider myself lucky. I came into this project relatively late, and my former PI, Weimin Ye, who's shown here actually in China at a meeting eating crayfish with all of us. One of the benefits of this project was that I got to travel to China a couple times and see field sites and meet people. And so Dr. Ye had this idea that he was going to do microbiome because he was really interested in it. And so he made arrangements that at one of the collection sites, we would extract the DNA for microbiome and for EBV. And we had that DNA that we were then able to do 16S rRNA sequencing, which is kind of like bacterial barcoding.
Right.
And we found two things that I am really excited about. So the first thing that we found was that there was a difference in the microbiome associated with whether or not you had cancer. And can I get on my soapbox a minute, Mark?
Please.
So when I work with microbiome data and I say that there's an association, it's a little bit, it's sometimes hard to articulate what is that association going to be, or what do we think the association will be. So if my colleague is looking at blood pressure, the blood pressure in her participants can go up, can go down, or can stay the same. And, you could maybe you can expand that out and say systolic, diastolic, but at the end of the day, it's up, down, or stay the same. I work with a microbial community, and so the diversity inside my sample might go up, might go down, might stay the same. The distribution of the microbes in my sample might go up, might go down, might stay the same. Specific microbes might change. And so breaking down, and so part of what becomes challenging, and part of, frankly, what I really love about analysis, is trying to put together a set of hypotheses of what, based on the data I can get, what specific thing is changing. So the first thing I did with this data set was just look at the richness. So the number of organisms that exist in a sample. And we found, and I was really trying to prove to myself that there wasn't an association. I walked in with the goal of, I'm not going to find anything, and I'm going to work really hard to show that there's nothing here. So when we measure the microbial community, we tend to measure big community aspects. one of the aspects we can measure is the alpha diversity, which is the within sample diversity. So it's the number of different types of things we see. If I go to my closet and I look in my closet and I have tennis shoes and high heels and flat shoes and sandals in my closet, I've got 4 different types of shoes. It doesn't matter how many pairs of tennis shoes or how many pairs of high heels I have, I can count up the number of types of shoes I have. And maybe we go to your closet, Mark, and maybe you have several pairs of Doc Martens, maybe you have a pair of tennis shoes, maybe you've got sandals, and we can count up the types of shoes in your closet and go, oh, Mark has 3 types of shoes. And we can look at that comparison and go, oh, Justine has more types of shoes than Mark does. Except that rather than counting types of shoes, I'm counting different microbes. We could also take our closets and compare them and say, well, Justine has this type of tennis shoe. Justine has a pair of Converse, and she has, whatever shoes she has. And maybe you also have a pair of Converse in your closet. And then we'd say, okay, we have one of the same type of shoe, but maybe we have different shoes. Can I take a minute and talk about my absolute favorite beta diversity metric?
Please.
And so I have a set of microbial communities. I usually talk about these as houses on my block or communities in my apartment building. And so I've got 3 communities here. I've got a teal community, a pink community, and an orange community. And you can see that they've got the same set of organisms in them, that they've all got humans, dogs, and fishes. And I can actually look at the relationship between those organisms in the community and say, hopefully very intuitively for you, Mark, and your listeners and viewers, that a human and a dog are more similar to each other evolutionarily than they are to a fish. And I can take that evolutionary relationship and I can compare the communities on the basis of that. So if I take my teal community and my pink community, where my teal community has 2 humans and 2 dogs, and my pink community has 4 humans, I can paint that back onto the phylogenetic tree and say, okay, I get the branch for the dogs. That's unique to my green community. And that's going to be my unique portion. And then I look at the whole tree that I have. So I have my humans and my dog. And I can count up how much evolutionary history is unique based on that dog and how much is shared based on that human and that dog. And that gives me a measurement called unifrac, or the unique fraction of the phylogenetic tree. And that is my favorite way to measure beta diversity right now.
No, it's a really good thing to talk about because you're looking at a really complex set of populations, and there'll be some things that are the same, some things that are different. Individual A may have a whole lot of one member, individual B a whole lot less of that same member. And how do you describe all of these things? And this is where the number crunching, this is where the bioinformatics comes from.
Absolutely. And this measurement won't, so the unifrac measurement I'm talking about today won't tell you if you have a whole lot more of one or the other. There's a different measurement I can use for that, a different yardstick. And in my analyses, I like to use a lot of different measurements or a lot of different lenses.
So we start to look at what lives. At first, you looked at saliva, and this enabled you to look at EBV and also whatever bacteria and archaea and presumably fungi are there. True?
We use so the analysis
16S,
I did
right.
used 16S rRNA, and 16S will only pick up bacteria and archaea. A colleague of mine, Yufeng Chen, and here's a picture of me and Yufeng in China with our other colleague, Yuwen, then did a fungal analysis later using a different set of primers that identify fungi.
So this is really interesting to start comparing the two groups, right?
Yes.
So, and this is just been based on things that have been collected from the saliva. Is that correct?
This is only based on the saliva.
Okay.
And I will also caveat, Mark, and make it clear that this is the saliva of people who already have cancer versus controls.
Got
so we don't know if what we're finding happened after the cancer developed or happened before the cancer developed. We started looking, and when I looked at alpha diversity, so that measure of the number of different types of organisms, I saw a huge difference between people who had cancer and people who didn't. And it was ginormous. I think my test statistic, so I put it into a statistical test, had a value of like 99. And the value of the next biggest thing had a value of like 9. And so it was just this massive difference between the two values. And I, who went into this process as sort of a skeptic, had to go, okay, maybe there actually is a relationship here. And so we looked at that. And then my next step is always to look at beta diversity, or this high level between community comparison. And when you look at beta diversity, I tend to make a map of the data. So here I've got a map of my data, and you can see it makes kind of a Nike swoosh. And I've painted on information about the alpha diversity. And you can see that it goes from like purple to yellow. And as it goes from purple to yellow, the alpha diversity gets bigger. And you can see that there's, hopefully by eye, that there's an association with that alpha diversity and the x-axis. I can also take this same data and paint on whether or not somebody is a case or a control. So the people who have cancer are shown here in red. The people who don't are shown in blue. And you can see less by the plot itself and hopefully more by the marginal axes that show you a distribution, that there's a shift between the people who have cancer and the people who don't. And that the people who don't have cancer are bunched a little more together further along that x-axis. Remember that that was our high diversity axis. And so we're seeing them bunching further to the right in that high diversity region. And then there's a broader tail of the people who have cancer. And so when I looked at that, the effect of having cancer was 5 times bigger than the effect of anything else I measured when I tried to come up with other things that would affect the microbiome. And so based on that, we had to go, okay, there's an association. It was hard to keep, pretending that nothing was happening. And after I see a difference in beta diversity, so a difference between the communities, I like to test for specific organisms that are driving that difference. And we used a couple different tests, but we kept coming back to these 2 bacterial barcodes. And the bacterial barcoding method we used gives us a single nucleotide resolution. So we can go all the way along the sequence identifier. And if there's one nucleotide position that's different, we can tell the difference between those 2 sequences. And we had these 2 sequences that kept popping up that we called GRAN 7770 and GRAN 5A37. And they belonged to Granulocatella adicans. And they were identical except for a single nucleotide at in sort of the V3 hypervariable region. So we found these 2 particular barcoded organisms that were in all but one sample. So we had almost 1,000 people, and out of 1,000 people, we had one person who didn't have one version or the other. And when we looked at it, we found that if you had one version, you were 1.5 times. So you were 1.5 times more likely to have cancer if you carried both versions of the variant. And then you were 2.5 times more likely to have cancer if you carried so If you had 5A37 alone, that was sort of the good variant, the good twin. If you had 5A37 and 7770 together, 1.5 times more likely to have cancer. If you had 7770 alone, 2.5 times more likely to have cancer than having that 5A37 variant.
This is so interesting to me because this is suggesting, you know how reductionist we all are. We want there to be one bacterium or one virus that does this important thing. And there are things like that, yes, right? But what we're seeing here is it's sometimes the interactions between things and the interplay between them that make the difference. And that's exactly what you were describing here. And that I find that fascinating.
One of the analyses that I felt so privileged to get to run, because I don't always get to run some of these fun analyses because the data and the hypotheses just don't support them, was to do a co-occurrence, co-exclusion network. And so here you can see the network that I brought with me. And the stars, the green and the purple star, are my 2 Granulocatella variants. And you can see that they bring some friends with them. And if you look at their posses of friends that are showing up with them, the colors tell you about the genus, and that they're bringing along a common set of friends. So they both bring along Streptococcus, they both bring along Veillonella, they both bring along Prevotella, but they're slightly different versions of the same genus that sort of like to hang out with the variant that you get. And so it's sort of like these were markers for a broader community. And actually, one of the analyses that we did was to take those back out of the community. So we pulled off the 5A37 and the 7770, and actually anything from genus Granulocatella, and we recalculated our unifrac distance. And when we looked at that second axis of our PCOA plot, the map I showed earlier, was associated with which Granulocatella you had, 5A37 or 7770. And so here you can see my same map that I remade. And you can, again, if you look at you can see it's colored by case control status. If you look at the x-axis, you can see that distribution of alpha diversity. And if you look at the y-axis, you can now see whether they have 5A37, both, or 7770. And I have never in my professional life seen a case where where you can define PC2 on a PCOA, the thing that explains the second largest amount of variation in the data, defined by 2 features in that data set. This was, I was like blown away and floored to see these 2 organisms that were essentially identical behaving in such different ways associated with cancer.
And it's funny because, funny. It's striking because, again, we're so used to thinking about one patient and changes that take place in one particular but this is a large constellation of people. So this means there's something really going on, and there's something, it means something. And this is I would certainly share your excitement in sitting in your shoes.
Well, I was super excited. I am still very excited about it. That's why I'm talking about it 4 years after I've moved on to new adventures. And I sort of wish that I could tell you that other people have replicated my work. And unfortunately, one of the challenges about this work is that the reason we could see those Granulocatella variants is because when you sequence the 16S rRNA gene, you use different primer pairs that target different hypervariable regions. And the primer pair we used targeted the V3 region and the V4 region. And that single nucleotide variation was in the V3 region. Subsequent studies have used primers that only targeted the V4 region.
Yeah.
And so they couldn't pick up the difference between our two granulocatellas. They couldn't see those ASVs. There is one piece of news that I sort of cling to, though, in that, which is that we saw our 7770 variant, our cancer-associated variant, was also associated with a streptococcus sequence. And that streptococcus barcode was a really abundant organism. And other people have done work where they've found a similar streptococcus. So I sort of have hope that even though they couldn't find our granulocatella, which I believe is sort of the, can I use the word keystone, Mark?
Sure.
Do I dare? Which I think is sort of the keystone organism, but they're finding somebody who's maybe a proxy.
Mm-hmm. I think that, so in other words, rather than if it's hard to identify the one particular organism, you have one that's easier to identify that co-occurs with it.
Yes.
And I need to tell the listeners and viewers that when you look at a 16S ribosomal RNA region, there are some areas that don't change very much and some areas that change a great deal over evolutionary time. That's where that term hypervariable comes from. Now, you of course know this. I'm saying this to listeners and viewers. So, for example, when I do the kind of work that I do with organisms that my students isolate, say from their water bottles, we're not doing the fine level work that Justine is talking about. What we're doing is just looking at gross differences. So this is why you can miss things so easily. And that's what, you must be so frustrated they're not using V3 primers, is what I'm trying to say in subsequent work.
I am a little frustrated. I also appreciate that microbiome studies take years to do. And so the work that I'm talking about was started in 2013. I think the extractions were done in 2016. I joined the lab and started working on the analysis in 2018, and the work finally got published in 2020. If there's another paper that comes out in, say, 2022, I assume that they started if their analysis is published in 2022, I assume it takes them 6 to 8 months to get it accepted, which means that they were probably analyzing their data in 2018 or 2019, the same time I was doing my work, and they probably did their sequencing before that. And so I find it frustrating that they didn't use the same primers, but it's also not fair for me to look at the literature and look at other people's work and say, I really wish you use this technique that you didn't know you needed to use. Or my colleague used to say, I really wish you'd use this tool I haven't built yet.
Yes. I think that there's nothing like retrospection. Everything looks better in the rearview mirror. You always have some idea of what you should have done before. And that's one of the challenges. And I just want to emphasize one more time to the listeners and viewers, the complexity that we see in any particular niche in the human body where you have lots and lots of microbes, and how they're constantly going to be changing and varying. So the way in which you analyze these things can be very important. What gives me pause is whether or not, if we don't see associations that are interesting, have we used the right set of primers to look for them? We don't know. Right?
I will go one more, Mark. I will say, have we used the right set of analytical tools?
Yes.
Because I can tell you other stories about looking at data, and I used a different set of analytical tools than someone else did. And I drew a completely different conclusion. Mm-hmm. And when I reanalyzed their data the way I analyzed mine, I drew my data and their data came to the same conclusion. And when I took my data and analyzed it the way they'd done theirs, again, and we were drawing opposite conclusions. So their analysis didn't find a signal. My analysis found a signal. And it was legitimately that we looked at identical data in slightly different ways.
Yeah. So let's talk about what this means for listeners and viewers with regard to this particular finding. What you can say is that from your work, you found a particular set of organisms that appear to be associated with the cancerous state.
Yes.
Okay. Now, those organisms, has there been any movement on I presume they can't be cultivated.
They can. They absolutely can be cultivated. They're normal oral microbes. The I don't know, joy and challenge of interdisciplinary work, and possibly a piece of my career trajectory, is that the people involved in the study would, one, have to go out and collect new samples to be able to isolate them. And our case control study was done 10 years ago now.
Right.
And I will say that the political and the research landscape in southern China, where this work was done, is certainly affected by the coronavirus.
Yes.
The people involved in the project are also not, I don't want to say not entirely, but are not wet lab microbiologists. Most of them are epidemiologists and clinicians. And so for them, the idea that you'd go out and you'd, ask somebody to give you a spit sample and then go back to your lab and grow plates is maybe not something that is comfortable or familiar.
Got it.
And so I also left the research group shortly after this was published and went to chase another adventure. And so I don't think that anyone has cultured the organisms yet.
So I think it's important for listeners and viewers to appreciate, now I am by no means a cancer biologist, but some forms of cancer appear to be, triggered is a strong word, but their likelihood increases under conditions of inflammation. So one could see perhaps a set of organisms that tend to be associated with inflammation. Therefore, in those people that might be expressing cancer, you'd have these organisms. So perhaps they could be used as something like a tag to let you know that this person might be in the process of developing or already has this form of cancer without other forms of analysis.
Yes, I think that is a possibility. I think that to get there, they will have to have a way to identify the organism. So maybe an organism-specific PCR primer.
Yeah.
And then you would go and you would screen a whole population of people, and you would follow them for, say, 5 years and say, if we detect this variant, do in 5 years they get cancer. I don't know that anybody has done that study yet, but if they have, I would be super interested to see the results.
So I'm going to post a link to that article that you've been discussing here for people to look at. I'm also going to find other links that discuss various aspects of how you look at things. For example, I know that you're using CHIME, so there's a link to CHIME as a bioinformatics tool. I'm going to put links up to a little bit of information I have on 16S, a little bit of information on where's my brain today? On alpha and beta diversity, just for people that are interested in following up on that. But what I'd like you to do, if you wouldn't mind, is can you tell the listeners and viewers, like what 2 things that you'd like them to take away from this particular study? Remember, some folks are what I call beginning micronauts. They don't know very much microbiology. Others are real experts. So I don't know, maybe one for each of them?
So I think my number one takeaway, and my number one takeaway for everybody is it depends. TM. And so the answer you get is going to depend on the question you ask and how you ask it. And
Right.
here, the answer we got was dependent on the part of the 16S sequence we looked at, and it was also dependent on the way we did a bioinformatics step. So our ability to find evil twins was because we were looking for a feature that identified the twins in a way that we could get that resolution. I think for people who are a little more experienced, I would say that this is a study that argues that 16S sequencing isn't a fossil the way I think a lot of people say. If I had done this with metagenomics, at least the way I annotate metagenomics, I don't know that we would have found those granulocatella variants, because in a, say, reference-based annotation, I just don't know that we would have found that difference. I think the signal we saw was a signal we saw because we had a large population with 16S. And maybe that comes down to being lucky. That I had the right primers in the right data set with the right technique, and everything came together to see what we saw and to give us this story.
Just remember, the OG microbiologist Louis Pasteur famously said, it's better in French, but I don't speak French, chance favors the prepared mind. So there you go. I mean, you can call it luck if you want, but it's also your skills.
Thank you.
So what I'd like you to think about, and I know we're starting to run a little bit short on time. You mentioned other adventures. Can you tell listeners and viewers, maybe in just a minute or two, what your current adventure is?
Sure. So I currently work primarily with the ECHO Children's Study, Environmental Influences on Children's Health Outcomes, big acronym, which is a multi-site, multi-center NIH-funded study looking at all sorts of exposures that affect children's health. And I have the unofficial title of Microbiome Chatelaine. And
seriously?
It's my unofficial title. I coined it.
I love that.
But I describe myself as the Microbiome Chatelaine. And for those who don't know what a chatelaine is, she was the housekeeper in medieval households, the sort of head female servant, and the title has specifically come to mean the keeper of the keys, or refers to the keys. So there's actually a device also called a chatelaine. And I've done a huge amount of bioinformatics. I help people access the microbiome data now that it's ready to analyze. And I get to do some analysis myself, and I am really excited to see what we're going to be able to do with our data set of about 15,000 samples from about 1,000 children and their parents.
To me, this is almost like a combination of drinking from the fire hose. You know, you get all this data shooting at you, but you can only catch a little bit at a time. But it's so much there, and it's almost like our ability to collect and analyze data has gotten ahead of our ability to actually get particular conclusions from it, because we don't have enough context yet. But it's not a surprise, because it's so complex.
I mean, I am a woman who chases large data sets around the world, Mark. I cut my teeth on the American Gut Project, and so that was my first introduction to the microbiome. It was early work on the American gut, and then I went to Sweden to work on big data sets in Sweden. And I came back to the US to work at Hopkins on this big data set. I just like big data with microbiome.
Yeah, you like big data and you cannot lie, right? There it goes. I'm sorry about that, but it's funny. But this has been wonderful. And I'm so impressed. It's so hard to talk about the realities of this kind of analysis. And what I think a real value of what you've done here today for the listeners and viewers is shown you the challenges and the promise of these approaches. And so someone might say, oh, it's just too complicated. Well, how do we get past that? By doing exactly what you're describing and bit by bit chipping away at things until we get to learn what the real patterns are. And we're so used being reductionists to look at one particular thing under one condition, but that's not how life is. And that's not how we're going to answer the questions we face now. It is how we answered certain questions 100 years ago. It is not how we answer in the future.
Absolutely.
So I really want to thank you for this opportunity to chat a little bit. I love your evil and good twins idea. That's just amazing to me. I love it so much. And we haven't even talked, and I'm not going to make you talk, about the herring microbiome diet. We're just going to stay away from that from now. But I'm going to put a link to that up for people. Maybe that's going to be enough tempting to get them to read that. So the effects of the diet on the microbiome, even under very unusual circumstance, I'll bet that listeners and viewers will be interested in that. So I think we've learned what you really love the best in the microbial sciences is analyzing those giant data sets. And I think we've all learned a lot today.
Well, it has been a pleasure to be with you. Thank you for letting me come and talk about my work.
Oh, it has been such a pleasure. Thank you so much, Justine. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Justine Debelius is in the Department of Epidemiology of the Johns Hopkins Bloomberg School of Public Health in Baltimore, Maryland. Many thanks to David Renata for superb editing, and Reber Clark for the wonderfully quirky music that brings a smile to my microbial heart. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.